My name's Bob, says I'm from the Datta school. I want to start with like two confessions. The first is that I went to a meeting with Venki. Venki had a great time, and I got super sick. So I'm not going to be like the bubbliest person, but I should be okay. The second thing, which is the more important confession, is I don't know any math physics, really. I'm going to talk a lot about some behavioral modeling, which I understand conceptually, but if you ask me to dig in too much, I'm not going to go into those sort of things. But everything that we do is in collaboration with my friend and colleague, Scott Linderman, who's at Stanford, and has really been instrumental in the last decade of my lab's work in building the models that I'm going to describe for you today. So today I'm going to talk to you, I'm going to give you kind of a quick overview of stuff we've done in the last 10 years or so. I'm going to talk a little bit about work by Jeff, Lynn, and Maya over in the lab, looking at fast neurobehavioral relationships. I'm going to talk about some very recently published work by a postdoc in the lab in Caleb, looking at slightly longer time scale relationships. Then I'm going to talk about a bunch of unpublished work on how behavior evolves over the lifespan and across evolutionary timescales. Okay. So yeah, let's begin. So my lab has been for a long time deeply interested in this problem of natural behavior, which I think is something that many of you are interested in. Now, how do animals generate complex patterns of movement in the wild that are self-directed and self-motivated and reflect the animal's ongoing needs and desires rather than being imposed upon animals exogenously by experimenters, through overtraining or the imposition of motivations and drives. And one explanation for animal behavior, this kind of natural self-motivated animal behavior, arises from ethology. The ethologists working 75 years ago thought a lot about these problems. And they hypothesized that ultimately one way in which natural behavior addresses the control problem is to modularize behavior. They hypothesized that natural behavior is built out of repeated motifs or modules of movement, stereotype, brief, three-dimensional bits of action that the animal can string along into a variety of sequences flexibly and organize hierarchically over time, thereby imposing upon natural behavior a kind of meaning. And so we have really embraced this kind of idea that behavior is built out of models. We studied the mouse. And so I just want to give you a quick sense of how we got into the idea that mice might actually, in the lab, might actually organize their behavior out of modules. And our ideas came really from simple observations. So this is a mouse in a bucket I bought from Walmart, and you can see it's running around. If you watch this movie, for some of you, but not all of you, have seen mice behaving. Never seen mouse behave? That's what it looks like. It looks just like this. And what you'll see as you watch it is that you as a human have some words for what the animal's doing. It's running around, it's stopping, it's grooming, it's pausing. Another thing you'll notice is the mouse is kind of fidgety. Like it tends to do a little thing and switch to another thing. And so there's a kind of, and it also seems like the mouse's behavior is very much non-random. That it seems like it's exploring this bucket and it's doing so in some sort of organized pattern between pausing and grooming itself. Okay? Obviously, a lot of what the animal is doing is not well captured by this 2D camera. And so often, but not always in my lab, and for most, but not all the data I'll show you today, we actually switched to using 3D cameras, which is just a single camera above this bucket that sees in 3D. So here he's mapped over this mouse. This is height at any pixel. And the pixels are red, it means that part of the animal is kind of high up towards the camera. And our first real insight just came from a really simple analysis of these data. We took that movie I just showed you. We isolated the mouse, realigned it along the axis of its spine. Its nose is over here and its tail is over here. And then we used random projections just to compress this data down literally onto this axis. And we played the movie over time. And what you see if you watch this or any number of movies that are like this is that there's a kind of self-organized block structure to the data where the animals can be behaving and you see that the data are perforated by these lines, right? And these lines happen on average in the mouse about every 300 milliseconds or so. In the rat, it's about every 600 milliseconds or so. And if you do something that's block here, and if you kind of line up what the animal is doing during the execution of one of these blocks, it looks like a little motif action. Okay, so like So you might hypothesize in your head that like this block is the animal rearing up, maybe this block is the animal turning to the right, and so on and so forth. And so it seems like from just inspection and then a bunch of model-free analysis, that there's a kind of rhythm to mouse behavior, a kind of hyper-illustrating rhythm that's exposed by 3D data. And so we wanted to kind of capture that. And so we've built this system called motion sequencing, or MoSeq for short. And MoSeq takes as its inputs either depth data, like I just showed you, or key point data that I'm sure many of you are familiar with, which is a way of tracking individual points in 2 or 3D video on animals as they really behave. And these data are then fed to this motion sequencing algorithm, which is really a hierarchical modified hidden Markov model, where each little behavioral module, or we refer to as a behavioral syllable, this model is an autoregressive process through some high-dimensional pose space that's captured by the data. And switching dynamics between these little modules are imposed by a modified Markov model. We fit these models with kind of Bayesian non-parametric techniques, and that allows the structure of the data to essentially reveal to us the optimal number and identity of behavioral syllables in any given behavioral data set. I'll mention that the idea of using a Markov model to model behavior is kind of quaint, right? It's very, very old. I'm sure many of you are interested in much more predictive models that take advantage of deep learning or language models to kind of forecast future behavior. And these are really powerful methods that are part of the future, right? We have chosen, at least for the data I'm going to show you today, to kind of use these relatively simple methods given modern techniques, primarily because they capture the thing that we care about most, which is modularity in behavior, and also because they're super interpretable. And as I'll show you, my lab is particularly interested in understanding the neural basis for natural behaviors, and these modules do quite a good job at capturing temporal discontinuities in neural data that have explanatory power for the behaviors that we're observing. Okay.
Can you say something about what the transitions are in these modules?
Yeah, so the transition, I'll show you at the neural level, but at the behavioral level, they're basically, you can think of, when you think about what a mouse is doing, often the mouse will take a few steps during a run, it'll stop, so that's a transition. It's like, you can almost think of the transitions as being well quantified by jerk. Like basically whenever there's an abrupt transition in behavior, which mice exhibit all the time, every 300 milliseconds or so, that's what one of those transitions is. So the mouse rears up, that would be a block. It rears down, that would be another block. And that's just because the mouse has these discontinuities naturally in its behaviors that are captured by it.
Can you unpack that jerk in terms of whether it's the jerk associated with the motion of the entire body, or whether it's motion of maybe a limb or head?
So that's a great question. When we use the depth camera, we treat all pixels as equal. So we use some dimensionality reduction. So in that case, we don't. Typically, we believe in what we know, that we're really capturing the overall body pose as dominated by the spine. When we have key points, we have the limbs, and we have the whole body, and we can do exactly the analysis that we suggest. And what that reveals is that, of course, there is quite a bit of nesting, where the dynamics that you observe in the spine aren't necessarily, and the breakpoints you see in models built out of spine-only data might not correspond directly or clearly to what's going on in the limb.
And are there also frequencies of these which are function of size? In other words, you'll have many more maybe jerky movements of the finer motor parts and much fewer.
Absolutely, yeah. So for example, the thing that my lab is particularly interested in is smell. And so when animals rear up and they are actively sniffing, those sniffs can be 10 to 12 hertz, which is much faster than this 300 hertz time scale. And so in our published work, we show that these models do a good job of capturing, can be used to capture kind of all these time scales, depending on what you're interested in. And so an important point about these models is, they're basically unsupervised data-driven models, but you do have to provide the model one time scale. You have to tell it like, oh, I believe that the thing that is most interesting in this data is at the sub-second time scale. For example, if you look at posing, or is that the 100 millisecond time scale if we're looking at sniffing? And we've done photos. Yeah.
you tell which changes come from the structure of the environment and which are come directly from the mouse?
Yes, I can. None of the changes come from the structure of the environment because always we dump in a $2 bucket. So the animal's in the dark. And so the only change actually the animal is capable of exerting on its environment is pees or poos in the bucket. And animals love to come back and sniff those spots. Actually, they use them to organize. They often, people think, can organize the geometry of a otherwise featureless environment. And so that's probably the only.
What about the walls?
I'll talk all about that. So when we think about the structured behavior, it turns out that even in an empty bucket, there are affordances, and animals will take advantage of them to structure their behavior. So there's a whole, I'll talk about that. Any other questions? You good? Okay. So we have this model. It tells us how many syllables there are. It tells us the duration distribution for each syllable. And of course, it tells us the order in which they occur in any given experiment, which we can then kind of synthesize into these state maps, which I'm sure you're familiar with. Here each note is its syllable, its diameter, how often it's used, the thickness of these arrows reflects the observed frequency of transition from one syllable to the other over time. Fairly straightforward. And we've been super interested. Just going to get my wired. So, and we've been, really interested in trying to understand this structure. It's clear that the brain is articulating rules that govern how often syllables are used. Right, some are used a lot, some are used a little. And the brain's also articulating rules that govern the order in which these are unfolding over time. And we've wondered where those rules come from. For reasons I won't explain to you, we've hypothesized that a part of the brain called the striatum, and in particular, a subregion of the striatum called the dorsolateral striatum, acts as a kind of control node to create this structure. And to test that hypothesis, we've been doing neural recordings where we put cameras or electrodes in the dorsolateral striatum as we do motion sequencing, and we align the data and we look for neurobehavioral relationships. So this is one such experiment. Here's the mouse skin that's been aligned. Nose is here, tail is here. The cable coming out of this mouse's head. This is a camera in the dorsolateral striatum, allowing us to assess single neuron activity as the animal fluid runs about. Post hoc, we can align the activity of each of these neurons with the ongoing expression of individual syllables, and we can ask if there's any relationship. Okay. And the first thing that we find, which I think is really exciting and tells you something about the reality of the idea that behavior is organized at the sub-second time scale in the mouse, comes from just aligning the data. So here's time, here's activity. Time 0 here marks the time that animals begin to express each new behavioral syllable as identified by Mosi. What you see is that systematically, there's a burst of neural activity in dorsolateral striatum. So there's a hyperultradian neural rhythm that matches the behavioral rhythm that I've been describing. Okay? And this activity is the consequence of a new neural ensemble turning on every time the animal expresses a new behavioral syllable. And it turns out that the identity of the specific neurons that belong to that ensemble do a terrific job of predicting which 3D syllable the animal's going to express at any moment in time. So there's about 50 of these syllables in total that an animal expresses in the output field. And you can use these neural data to make extremely accurate predictions about the animal's overall 3D pose dynamics as it behaves. Yeah.
So can you just comment on the delay between the transition of syllables?
Yeah. So here, this is just an indicator delay. It's very short. It's because calcium is slow. We've done exactly. So we've done recordings with this is older data, but of course at this point we've done many recordings with NeuroPixels probes, and we think basically the change in neural activity precedes the transition for sure, and then unfolds through the transition. Right? And so because this is calcium data, it's a little bit slowed down, and so if you take the derivative of this, it clearly precedes it, which is a standard way of kind of accommodating it. Okay, so it's clear that the striatum contains within it a code for the animal's 3D behavior. It's also clear, if you just do some analysis, that there's a little bit of information about the order in which the syllables are occurring over time that's held within the striatum. And if you lesion the striatum, it turns out the animals express all the syllables, they're just all in the wrong order. And that means that if you do simple things like startle the mouse or give it a smell it's supposed to run away from, like a box odor, it can no longer execute those behaviors. So it means that this structure is coming from, at least in part from the dorsolateral striatum, but the experiments that I've described to you don't answer the questions that they posed, which is, why is this expressed a lot and this a little, and why are the syllables in the sequence in which they are. And so we fall.
Can I ask a quick question? In previous slides, I guess I've seen this data before. It didn't occur to me that is that ramping up prior to the transition real? And if so, is there some evidence that you're kind of building up to that?
Yes, it is. So these data are a little bit, it is real. These data are a little bit too noisy for us to know much. I will just tell you, which I'm going to show you data, but we have exciting new data where we've done simultaneous recordings of motor cortex and striatum. So motor cortex talks directly to these neurons in striatum. And it's clear that there are planning signals that are present in motor cortex during the execution of the previous signal that are predictive of the specific behavioral choice the animal is going to make. And so during natural behavior, the same kind of planning signals that Mark Schurschland has seen during learned tasks, those planning signals are evident to us through the segmentation of behavior. Okay, but good on.
Can I just continue that? Plus, if I look at two-point correlations before the event, so to speak, do you see evidence of a decision, or am I overreading?
Sorry, what do you mean by two-point correlation?
So if I look at time before 0, and I look at 2 points before that, and then I just take any 2 points, T1, T2,
Okay.
and I keep the gap between them the same, but I move closer and closer to 0. Do I see anything interesting in the correlation that I would have between the data? I'll continue this afterwards.
Yeah. I mean, it's clear that from a neural perspective, is that what you're asking? Yeah. So within a syllable, it's as if animals are taking a trajectory through neural space that then abruptly changes in neural space as they transition into the next
And
level.
I'm asking whether the noise starts to become colored as a function of distance to 0.
I think that answers yes, because there's this finding signal. Anything else? Yeah.
This tell you that there's something that's special about the time scale that you put into mostly just from watching the animal. You mentioned that's something.
Yes. And actually, I'll take. I want to get back to that in one slide, but yes, I do think there's something special, and I think there's an interesting explanation for where the time scale comes from. It's not a made up. It's not a made up time scale. I believe it's a biological time scale. Okay, so to begin to answer those questions about the nature, like why some are lost and why there's a certain amount of ordering, we've been looking at the main neuromodulator that influences the striatum, and that is the neuromodulator dopamine. So we've made simultaneous neural dopamine recordings, and mostly just like we did neural recordings of the primary neurons in dorsolateral study. And what we see here, so you can see here just by eyes, there's a ton of fluctuations in dopamine in the dorsolateral striatum. The animals run about in the absence of a task and in the absence of a reward. And it turns out that there is about one fluctuation here per syllable, and actually it's a fluctuation in dopamine as animals enter into each syllable. And after a lot of analysis and work, what we've learned is that these fluctuations in dopamine seem to be acting like an endogenous reinforcer, such that if during the expression of a given syllable there's a lot of dopamine, over the next few minutes, the animal will do that syllable a lot more. If instead there's a little bit of dopamine instead of a lot, the animals will do that syllable a lot less. And if we take just this one-dimensional signal and build a Q learning model, an RL model, to try to predict behavior, we can actually do a pretty good job of predicting the actual sequence of syllables the animals can execute, over the course of the experiment. And so what this tells us is that a lot of the structure that we're seeing here probably is really due to fluctuations of dopamine and neural activity in the dorsolateral striatum, which seem to be causal for building this map. Because if we manipulate in a closed-loop manner syllable associated dopamine fluctuations, we can change the structure of this map in a predictable way. Yeah.
Just clarification, in the RL model, do you take the dopamine signal as a proxy for reward? Okay.
It's exactly what we do. So if you treat this like a reward like signal, it ends up to give you the feeling that it's shaping a huge amount of the variance in the overall behavior. Okay, so this gives us the sense. This gives us the sense of a couple of things. One, the fact that this is happening gives you the sense that there's something decision-making-like going on, where the animal is choosing its particular trajectory through the behavioral space at a subsecond time scale. The other thing, of course, all this gives you is the sense that we have something of an understanding of how the structure is built. But there's something super unsatisfying about this description. And what's unsatisfying about it really is that while we can provide some sort of explanation for why the animal might at a given moment do this and not that, we don't really know why. Like, we don't understand at a higher level at what the purpose of these particular sequences is in any real sense. It also, MoSeq also fails the kind of smell test. So when we watch mouse behavior, often what we see the animal do is execute goal-oriented behaviors that lasts seconds, not milliseconds, right? So each syllable is 300 milliseconds. But like you have a mouse here in this bucket with a couple objects. What you see is that the mouse will take a couple seconds to walk over here, will explore this for a couple seconds, get bored, take another couple seconds to walk over here. And you can think of its behavior as kind of organized into these little bouts that tend to last seconds, not hundreds of milliseconds, right? That's just the vibe you get. And so we wondered whether that was true. That was like a true thing. Is there really another time scale to mouse behavior that's not at the millisecond level, but maybe is well captured by seconds? And so one way you can look at that, or Caleb looked at that in a more formal way, is through mutual information, which here is just asking how many syllables in the future or the past can you predict given a present syllable, okay? And so if you look at the actual data, so you train up a MoSeq model, then you have a bunch of labels, and you ask, given a given syllable, how predict are things in the past or the future? That's this red line here, and so it's pretty long. It's tens of seconds. MoSeq is a generative model, right? It's a Markov model, so you can train up a model and then emit from the model a synthetic syllable sequence. If you do that, the mutual information falls off about 5 seconds. In about 5 seconds. That makes sense. We tell MoSeq that it should look for sub-second architecture and behavior. So each transitions on this 300 millisecond time scale. And so obviously the models know anything about longer time scales. So its ability to model behavior and spit out sequences, falls off very rapidly in time. But the actual data, have much more structure than that. And so Caleb decided that one way we can fill this temporal gap is to add a third hierarchical layer to MoSeq. So there's this autoregressive layer, there's the Markov part, and then he decided to add another layer on top of that, where he would have a layer that specified a unique transition matrix for syllables within what he called a state. Okay, so he calls this model SMOSeq, which is state-based organized motion sequencing, where there are these high-level states, each one of which essentially is comprised of a unique transition matrix, which is learned from the data. And so you can, and this transition matrix within each state controls possible sequencing of syllables. So it's just a hierarchical extension of a Markov model. So nothing too crazy or too special. Although you need a lot of data to fit it because it's hierarchical.
Right. Is it possible to color this or somehow visualize this differently in terms of spatial scale, spatial organization, meaning fine versus coarse, whole body versus limbs or head and so on and so forth, to understand the structure of how these movements are put together? I'm sorry, I'm not particularly.
No, I understand kind of what you're getting at, which is that what you're expecting in your head, right, is that an animal has a kind of hierarchy of timescales of behavior that correspond to fine movements and increasingly coarse movements. Okay, I just want to emphasize, these high-level states are not composed of a distinct number of syllables, like you'd expect if there was a hierarchy of movements the way you're describing. Here, each state is its own transition matrix, and what that means is that every syllable is reusable in each state. Right. So I understand what you're asking, but that's not what this achieves.
Okay, so that explains what I was going to ask, because you probably said it before, but I didn't catch it. So the higher levels, you're swapping in different state transition matrices.
Exactly. So one thing that has been done in the literature ad nauseum is clustering.
Those are huge matrices, right? So
yeah,
there's a lot to learn.
It's a lot to learn. Exactly, and we have some tricks for simplifying.
Okay.
So that it's learnable because it's hard to fit, right? The thing that's been done a lot is you identify some modularity in behavior, and then you compute something kinematic about each one of the modules or syllables, and then you cluster based on kinematic similarity. So what's present in the literature largely in this space is if I have some syllables, let's group all the rears together and call that a state. Let's group all the runs together and call that a state. So this is very different from that. And this is important because we think that these states actually, as I'll tell you, represent goals. The animal, self-directed goals that the animal has. You'd imagine that when you or I are engaged in a goal, we often use the exact same movement, just in a different context. So it's important for all the syllables to be reusable across the states.
I see, and there's some sort of a maximum likelihood thing where it's sort of, if that matrix is no longer doing a good job of predicting the transitions, then it's not.
Yes, and the one tricky thing, right, is that because of that, our certainty about state transitions is not as precise as our certainty about syllable transitions, because you have to observe a few syllables to know that the transition matrix is shifting. Makes sense, right?
Yep.
Okay. So what are these states? Like, what are they? So it turns out, so we've watched a lot of movies and we've looked at these states a lot. And it turns out the states seem to correspond to the animal engaged in self-directed goals that it might have, or engaging in self-directed tasks that it has in the open field. And so in an empty bucket, the things the animal has to interact with are the floor, the walls, and itself. And so what emerges are states that last 10 to 20 seconds long, where the animal is circling in one direction, circling in the other direction, grooming itself, or investigating locally. That's what you'd expect. If we put a conspecific, what emerges are a bunch of social states where the animal now does all four of those things, but in addition, now there's another affordance for it to interact with. And so Shimoseek will capture those social interaction states. We put objects into the arena. Shimoseek will capture different object investigation types of states that wouldn't be present in the absence of that affordance. And this superficial observation gives us the sense that really what these high-level states are is the animal self-assigning itself tasks or goals in much the same way that experimenters assign animals tasks in traditional computational or neuroscience. Okay, so just quickly, I just want to stress that each one of these states is not strictly defined by the expression of a particular syllable or syllable sequences. So here, this is like a grooming state. Animal expresses the open field. You see the animal expresses grooming syllables a lot during the state, but not exclusively. And so Shimoseek is capable of understanding the animal as it kind of in some sort of egocentric mode, and is doing that continuously through time, despite the fact the animal is not actually engaged in like a characteristic grooming behavior. Similarly, for these two pigmataxis states, the animal goes left or right. If you look at how far the animal is from the wall, which is the characteristic thing that you think about during pigmataxis, you see that the animal's distance from the wall can vary quite a bit during the state. But fundamentally, the animal's behavior is driven and organized by its desire to be engaged with a particular wall, and thus, regardless of distance, Shimoseek will identify this as a state. Similarly, if you look at social states, obviously, in most computer vision tools today to understand social interactions depend on physical proximity. You take two mice, you measure how close they are to each other, you say, oh, that's a social interaction. So Shimoseek basically infers that two animals are socially engaged because one animal's transition matrix is dependent on the other animal. And it will identify that state basically independently of the actual distance the two mice are. So sometimes the mice are very close, and sometimes in this case, they're separated by more than half of the arena distance. And Shimoseek still knows that this animal's transition matrix is well characterized by being in some state in which it's interacting with a partner. And so this allows us to capture social interactions at a distance.
So you're saying that the kinematic variables are showing this up after the fact is showing that they.
Yes, exactly.
Okay,
That's exactly right. So in other words, these things don't define these things, right? They're a little bit entailed, but they're not definitional, which is unusual.
What can you clarify? So this particular version of Shimoseek was done with the key point?
This was on we've done it both with depth and.
I see. So this is it's still a Ma's question about it's not getting the very fine scale things. It's like there's some
That's right.
primitive spatial
That's right. Again, so here again, we're looking mostly at whole body movements at a 300 millisecond time scale. That's how this is defined. Okay, and so that's what the syllables are in this case. Okay. Shimoseek solves this problem of this gap, and you'll notice these lines match up pretty nicely. And that argues on the surface that adding one more hierarchical level is sufficient to explain the temporal correlations during a 30-minute open field experiment. So that's pretty cool, right? Because it suggests that if you explain behavior at the sub-second level and at the second level, that's enough to capture temporal organization in a half an hour to an hour long experiment, right? So there's only these two layers, basically, these two temporal layers. And we have an independent kind of confirmation of that if we build a behavior GPT by looking at sequences of syllables, feeding them in to an LLM. The predictions that we get go to chance at about 20 seconds. And so from these hour-long experiments, and so that again is kind of independent validation, kind of an MLH validation that this is the limit at which behavior can be predictable. Sorry.
So I'm just, thinking about the fact that, right now, basically with this Markovian evolution of these longer lived states, you predict the tool distribution of it a bit better.
Mhm.
Does that somehow imply that these longer timescale sequences of solutions are actually more principal limits of behavior somehow?
More, I don't know if they're more principled. Why do you say they're more principled?
Well, I don't know, in the sense that once you know them,
Yeah.
then it's enough to make a Markov model that predicts.
Yeah.
them.
I mean, I think,
So in that sense.
or the way I would say, I view it as the converse, right? I would say that I think the thing you might imagine that in a half an hour or an hour long experiment, is that there's state variation in the animal, like they get hungry or bored, or thirsty, or want a social partner. And it seems like none of that is really affecting what's going on, right? It seems like these seconds long states are enough. Yeah.
Can I come back to this separation of time scale, or potential separation of time scale, and ask, maybe you said this and I just missed it. Fine motor movements also occur fast.
Mhm.
And so is there an interesting way, maybe you've already done this, of filtering in space and time so that as you blur more and more, you're able to essentially capture the coarse scales, and then you orthogonalize those, and then you capture the fine scales gradually. And so you're able to sort of get motor vocabulary in addition to
Yeah.
what you already.
Yeah, so we've done a little bit of that
Yeah.
by showing that if you basically take a slider on the timescales, and in the fly, we've done this, and move it this way, basically you can identify essentially motor primitives that are very fast. And then you can kind of identify different scales at which the primitives add up into something that's a little more coherent.
And what I'm asking.
But I want to, yeah, okay.
Go ahead.
Go ahead.
I was gonna ask whether you can play with space and time simultaneously.
Yeah, we haven't done that. We've only played with time. Okay, so how do we know that these longer time scales are real? Like I showed you a bunch of neural data, right, for the short time scale stuff. If you're interested, like for us, how's this not an ML artifact, right? Even though it's predictive. So one way to do that is to look at the brain and where tasks and goals live is the prefrontal cortex. So we've done recordings in PFC during all of this. And what you see, interestingly enough, again, is as animals enter into these high level states, you see ensembles of neurons that are dialed in just by eye, seem pretty low dimensional. It doesn't seem like there's a lot of these ensembles. It seems like most of the activity in prefrontal cortex is related to which one of these high level states are in. And it seems like they're switching dynamics. All that seems to be true. And in fact, if you go ahead and embed the neural activity, so here, each point is a pattern of neural activity at a particular moment in time. If you then color in the points that are associated with each one of these higher level states, you see that this map is segmented, which means that you can just take neural activity in PFC and make extraordinarily accurate predictions about which state the animal is in. Because it's easy to figure out when the animal is here, it's grooming, when the animal's here, it's socially investigating. Does that make sense to everyone? And there is, again, I won't show you the data, there is again a rhythm of burstiness in which there's a new ensemble that seems to be associated with every state transition. All right, good. Okay, so there is one kind of problem with this claim that I just made, that there's something about PFC and this neural activity that reflects these states. And the problem is that there's a lot of data in the literature from folks like Ann Churchland and Matteo Carandini and others that suggest that basically movement is encoded everywhere in the brain. So wherever you look, you get some information about movement. And so if each state ultimately is its own transition matrix, the movements that an animal generates in each state are going to be different. And so then maybe like what this neural activity is just a trivial representation of movement that distinguishes the states, and there's no actual state representation in PFC. It's all
Later was that if you did something really trivial, like look at the power spectra of each leg movement, or the head movement, do a similar, like, my dumb
Exactly.
thing, and then I map that onto this when I see
Right.
something similar.
Right. So we've done that dumb thing. That answer is no. We can do it, but we also have an actual explicit comparator because I've showed you DLS data before, and I've claimed that the DLS seems like it's encoding information about these syllables, these fast syllables, right? So we can basically compare our ability to decode fast syllables, which are a surrogate for kinematics, kinds of things you're talking about. And we can compare the ability of DLS, which is here in blue, and PFC, which is here in red. You see that DLS does a much better job. Conversely, you can decode what state you're in PFC, but not in DLS, but not in DLS. And so there seems to be a hierarchy of essentially timescales at which behavior is decoded. But PFC is optimized for longer timescales, and DLS is optimized for shorter timescales. And the codes for behavior that live in those two areas are distinguishable because of this timescale difference. And so that means that I can't really look at PFC and see a lot of kinematics. I can't like decode the animal's ongoing velocity very well, or its moment-to-moment movements, but I can tell you that it's in a grooming state. And conversely, in DLS, I know that it's turning, but I don't actually know which state it's in. Yeah.
Yeah, you showed beautifully that dopamine can enforce this sort of lower level behaviors. Can you do the same to the higher level states?
With respect to dopamine,
Yes.
we're working on that. It's there are dopamine fluctuations in PFC. Those PFC fluctuations seem to map onto some of the transitions in states. A good question. I think we're thinking. Okay, how am I doing my time?
Yeah, very good.
You have 20 more minutes.
Oh, great.
That everything at home.
Great. That's
A lot of questions.
no problem. Okay, so we wanted to pressure test this idea that these states represent self-directed tasks or goals. And one thing we know about the prefrontal cortex is that when an animal switches from one goal to the next or one task to the next, what happens is that neurons and neuronal activity that are relevant to addressing the task at hand are emphasized. Okay, so when you look at activity in PFC, you kind of get a snapshot of what the animal cares about at a given moment, based upon what kinds of features about the environment or the animal are being emphasized at that moment. And so we wondered whether or not we would see this in our data. And so we asked about our ability to decode two variables that are critical to the act of stigmataxis, the act of following a wall as you circle in one or the other direction. The two variables that matter are what direction the wall is with respect to you and how close you are to the wall. So if we ask how well we can decode these two variables from PFC during states, we see that they actually differ. That we do a great job of decoding these variables during stigmataxis itself, and a less good job in these other states. And it's not like there's a different, that these decoding differences are a consequence of differences in the distribution of the animal's position with respect to the wall during these states. We can normalize these data and only take those moments in which animals are grooming at a particular distance from the wall. And then we can ask how well wall direction and wall proximity are coded compared to the exact same position in the animals engaged in stigmataxis, and we still see this improving coding. So that suggests that when an animal is engaged in a particular task, the variables that matter are more emphasized, consistent with the idea that this is some sort of goal or task the animal is engaged in. And we can actually further pressure test that by making stigmataxis even more important as a goal to the animal. And the way you can do that is by taking an angry mouse and throwing it in the bucket with the mouse that we're reporting from. The angry mouse will attack our mouse. And then after that attack, our mouse will freak out and get close to the wall and run really fast along the wall, trying to protect one flank and trying to escape from the attacker. Okay, so all of a sudden, stigmataxis is salient. And when it becomes salient, you see that these codes for direction and proximity become even stronger. We can play the same kind of game with affiliative social interactions or with novel objects. In all cases, when the animal is in the appropriate state, we see that the variables seem to matter to that state are really highly encoded. That's consistent again with this kind of somewhat controversial, I would say, but interesting idea that during natural spontaneous behavior, animals aren't goalless. They actually have goals. They're just not goals that we specify for the animal as experimenters. Is the PFC actually causal for any of this? If we lesion PFC, it turns out this long timescale structure and behavior actually diminishes. And it diminishes because the animals rewrite the distribution of high-level states that they're actually entering. So the sequencing of those states changes as a consequence of lesioning PFC. That's similar to the idea that PFC is playing some sort of important causal role in structuring the animal's long-term pattern of behavior.
That's a good question, Bob. So in the transitions in the PFC population activity, I know it's a skeletal imaging. Do you have a sense they're sort of abrupt switch-like, or is it sort of gradual switching into different states?
Yeah, so there's two things. So I kind of didn't want to talk about the details, but there's an interesting thing that's true about the PFC activity that's not true in DLS. Someone asked whether or not the neural activity preceded, followed the transition. In DLS, it precedes the transition. And that gives us the sense that activity just before the transition is causal for the choice the animal makes. It turns out in PFC, in every case we've looked, activity actually follows the transition. So the animal switches behavioral state, and then PFC reflects that switch. So we think of, I think cognitive neuroscientists often think of PFC as being causal to higher order structure, and everything we've seen suggests that's not true. We've actually taken data sets of others, like Weijia Hong has recorded from PFC in mice engaged in a tube test, where they're pushing each other and fighting, and he's argued for a causal role of PFC in success or failure of a mouse fighting in a tube. We take his exact data, we also see the PFC lags, doesn't lead these transitions. And that suggests that PFC is engaged in some sort of role where maybe it's defining the distribution of states the animal can adopt, maybe it's reflecting ongoing behavioral choices that are being made at lower levels in order to perform credit assignment or to allow the persistence of particular states. All those things are possible, but we don't think, at least in this setting, that PFC is actually some sort of master controller where your soul lives and all decisions flow from a top-down manner, which is a very conventional. Okay. So I think this is exciting for us because it suggests that we can begin to use, with the right tools and the right models, we can begin to think of natural behavior as being very similar to more structured behavior, insofar as we can now identify a kind of set of goals or tasks that the animal has. We can understand its natural behavior as being in service of some goal rather than just sensory motor or reflexive. I think is interesting. Okay, so given this plus what I showed you before gives you the sense that like animals have some free will, right? That like they're making choices at timescales of subseconds to seconds about what they want to do in the environment. They're deciding what syllables they're going to put into sequences, and you can see that in the dopamine traces. They're deciding what states they want to enter on the seconds on time scale. And you can kind of get that sense here, right? And we thought, so that's pretty interesting, that idea. And we also had another observation, which is from a paper that I'll summarize in one slide, which is if you take a male or female mice and you image them day on day for like a week, and you do a 2D embedding of their behavior, and you hold a decoder, you can really easily identify which animal you're looking at from like 5 minutes of behavior. So animals are highly individualized in the way they explore the open field. If you just like use syllable distributions, it's the thing that you're using to distinguish individuals, right? You get basically perfect predictions of who's who. And so that suggests that each mouse has a personality and is individualized in some way and is making choices for itself in a way that's stable. And we wanted to understand how individuality might emerge and might change over the lifespan. All right, so this was just done over a week. How does this change if you look at the whole life of the animal? So the two things we want to know is, the things that we can measure at the lifespan and age and sex. So how does that shape individual behavior? And what is the ontogeny of individuality in mice? There's a technical problem that we had to confront, which you'll appreciate, which is that as mice age and develop, their size changes. Male and female mice are different sizes. And if you have some pose-based model where you're basically taking some pose space and you're clustering behaviors in that pose space, differences in size and shape are conflated with differences in behavior. That's like a fundamental problem. And so we've created a bunch of neural networks that allow us to morph mice onto a reference 12-week-old male mouse quite accurately and can convince ourselves that this works. And that marginalizes out any effects that the model any artifacts that might be created in the model that's just relevant to size variation, and allows us to ask about actual behavioral variation. Okay, so what do we see? And this is the new stuff, and I'm like still thinking about it. So you just tell me what you think. So we started by doing a very simple initial analysis whose result was very surprising to us. We built a multi-layer perceptron that attempted to predict. So I'll tell you the experiment. Experiments, we took 20 male and 20 female mice, and we imaged them twice a week from 3 weeks old, from when they're weaned until they died. Okay, so it's a huge data set. It took 2.5 to 3 years of data collection. It's an enormous data set. Okay, for each session, you can compute like the distribution of syllable usage for 50 syllables, 60 syllables. We built an MLP that takes us as inputs age, identity, and sex, and attempts on a single held out session to predict the behavior of that animal. And it turns out this works great. We can explain 80% of the variance in syllable usage if we know who the animal is on a single held out session. We know who the animal is, its sex, and its age. That's why. And the thing that matters most is who you are. Your age matters a lot. Your sex matters some, but not the most. Yeah.
Can you give more details about what the outcome said?
It's a single session. So we take
Single session.
yes, we take a single. So that's really important. That's really important. So like the session, the model and the training has access to before and after, but it doesn't have just the single session. That's what's being. And so what that tells you is that if you have information that's local in time, right, about who you are and how old you are, there's enough structure in whatever that is to make extraordinarily accurate predictions about the way an animal's going to behave in an open field. This feels very much like animals don't have free will, right? It feels like the opposite, which I just think this result is the same. And so then the question is, where does all the structure come from? What does it look like, right? And so we can begin to analyze that. So the first thing we asked is just about age. So if you just look at the normalized use of, these are 50 different syllables. Here we're just going to heat map the normalized use of syllables, males and females over some subset of time. This actually the real data. This goes out much farther, but it's representative. You see that animals systematically rewrite the way they use syllables as a function of age. If you embed the data, you can see clear age dependent trajectories through a behavioral space. And these are such that, if you build classifiers, you can accurately predict based upon behavior alone how old a mouse is across its lifespan with a 2 to 3 week error of either side. So age plays an important role in specifying your behavior. And there's basically a quite deterministic trajectory, characteristic trajectory that all mice take through behavioral space as they age. And this trajectory is continuous. So we don't see there's a lot of evidence for, juveniles and young middle-aged and middle-aged and old and dying. We don't see a lot of them. There's just before death, there's like some stuff that seems pretty block-like because you're sick. But basically, it seems like there's this continuous age-dependent evolution of behavior that's characteristic of all mice in our data set.
The mice alone or not in groups? How many groups?
No. Here they're housed in groups, which is for ethical reasons. But here I'm just bucket imaging. So alone, there's no affordances at all, other than the wolves. Okay, which is really important because today basically all studies of mouse individuality are in larger social contexts where the individualization is driven by things like social interactions or the availability of affordances. And so because of that, the actual nature, like the fundamental nature of individualization is, I think, quite unclear. Here, we're just looking at like, if animals are left to their own devices with no external constraints, what do? And what you learn is that they take a very characteristic trajectory of space such that you can predict how old they're quite accurately. Yeah.
Yes, in detail on the experiment, but how do you normalize for circadian effects, right? Like the timing of the experiment during the phase.
We attempt, they're all inverted, right? And so we were doing them all, within a 6-hour window of each other during their subjective active periods. But it's not perfectly normalized. And so it's possible that some of the unexplained part of the variation is state stuff. But think we'll have little balance, right? Okay, so that's age. Sex is the same. It turns out that at sexual maturity, There's basically a step function where animals acquire stable sex differences. The differences are not enough that, again, with 100% accuracy, you can predict the sex of a mouse based upon its behavior anywhere across the lifespan. And here's where things get interesting. I just need 2 more minutes. This is the fun part. So here we're looking at individuals, okay, over the lifespan. As I told you before, basically across the lifespan, the entire lifespan, we can predict individual identity based upon behavior. So, right, so that makes sense. What's interesting is that if you look at these data, you get the sense that each individual mouse is taking a kind of fixed trajectory through behavioral space. And that is a super intuitive idea. You might imagine that maybe there's a critical period, like when animals go through sexual maturity, where their brain kind of crystallizes, animals become who they are, and then they take some path, some fixed path through the behavioral space that reflects their individual identity. If that were true, an animal's past trajectory should predict its future behavior, the individual level. So that's what you would intuit is going on. So, but that's, I think, not what's going on. So if you look at this, you can just think of this as distance between animals. So, if that's true, you expect animals to continuously separate from each other in behavioral space. What you see is that sometimes it's true, but it's not always true. We've created a metric that allows us, that basically summarizes how much an animal's present behavior reflects the kind of trajectory it was taken through behavioral space in the past. We call this directional persistence. And we see that animals have directional persistence on timescales of about 4 to 6 weeks, but they lose their directional persistence at longer timescales. So they're actually taking some path through behavioral space that's sticky, but you tend to do what you did yesterday, which is why you can make predictions if you take out a single session, but you'll lose that after that kind of tendency after about 6 weeks. So Caleb decided to model this. So he built a model. His hypothesis was, well, maybe individual variation is exactly the opposite of this kind of determined trajectory. Maybe it's a random walk. So he built a model in which each individual's model is taking a cumulative random walk. To accommodate the stickiness, he included a term in which at every time step, the animal's behavior is modified by some fluctuation that tends to regress to the mean, which allows you to kind of think about behaviors being sticky, and there's some noise. And if you take advantage of that model, you do a really good job of capturing the observed intramouse distances in our data set, and you capture these sticky but eventually random walk-driven dynamics in behavior. And so this suggests the opposite of what I started with. It suggests that at longer timescales, actually each mouse is being buffeted by random variation that shapes its behavior in this kind of sticky way. And it could be that the 6-week timescale is some sort of neural timescale that reflects an animal's memory. This could be some sort of drift tapping the animal's brain. But something about the animal's accumulated experience or noise in the animal randomly reshapes an animal's behavior over its lifespan. Yeah.
Can you comment on the ratio between the intra-individual variations of the segments? Is it comparable? Or is it
No,
it's for a single?
No, it's much smaller. It's much smaller.
It's not completely random.
Well, but that depends on how you're what's the time scale you're averaging the data, right? And so so there is so these random models do a good job over the long time scales of capturing this variation. So the 3 things you take away from this is the time scales, sub-seconds, seconds, right? It looks like animals are engaging in decision making because we have neural systems that are involved in decision making that are actually engaged, that are predictive of what's going on. The single session level, if you hold out 1 session, mouse behavior is extraordinarily predictable because mouse behavior seems really auto-regressive. It seems very sticky over this weeks-long time scale. But still, mouse individuality seems to be driven by kind of some random influence, which I think is super surprising. Because I think I would have assumed that they're taking trajectories through behavioral space. Instead, it seems like they're just buffeted about by random chance. And with that, I will end because I'm out of time. And I'll just say that I showed you the people who did the work at the beginning, but because I love the slides. But the work that on individuality has been driven recently by a guy who came to my lab with a pure math background, Caleb Weinrep. So it's been really fun working with him because he actually can model the random stuff.